Welcome everybody to today's webinar. Topic of today is an overview of the Orion Small Molecule Discovery Suite. Our presenter is Dr. David Hamilton, application scientist in the Boston office of OpenEye. And if you've got any questions in between, please fill in the Q&A section. We'll answer them towards the end. Without further ado, I'll hand it over to David. Please take it away.
Thank you, Gunter, and hello out there, everyone. My name is Dave Hamilton, and I'm an application scientist at OpenEye Scientific, now a part of Cadence Molecular Sciences. My particular area of focus at OpenEye is on applications to small molecule drug discovery, and as such, I'm here to give you an overview of the Orion Small Molecule Discovery Suite. The goal of this webinar is to give those unfamiliar with OpenEye and our tools a quick look at our solutions for impacting drug discovery, and to give current customers a look at the science that we've developed and continue to develop that leverages the Orion platform and its massively parallel compute architecture. But first, especially for our new customers, I want to give you all a quick look at who OpenEye is as a company and our overall set of offerings. OpenEye has been creating tools for computational drug discovery for 25 years, and our mission under Cadence Molecular Sciences has not changed. We seek to revolutionize computational drug discovery with physics-based, scientifically rigorous software aimed at advancing efforts to create new and better therapeutics faster and more efficiently. As such, we have created several layers of solutions for our customers, including programming toolkits comprising our proprietary code for cheminformatics and molecular modeling, user-friendly applications that leverage these toolkits to carry out common computational chemistry tasks like 3D similarity searching or docking, and now Orion, our cloud-native platform for molecular design. As the focus of today's webinar is on the science found in the Orion platform relevant to small molecule drug discovery, it makes sense to first give you an idea of what the platform is in order to help you understand how it enables the science that we've been known for over 2 decades, as well as the new directions that it's enabled us to explore. Orion is a fully integrated platform for drug discovery containing functionality for analysis of data, for viewing results of calculations in 3D, and for storing your data in a folder-based architecture. It also contains a number of features to enable collaboration across teams, including team-based projects, discussion boards, snapshot capabilities, and shareable link creation, in order to facilitate quick sharing of data views through any mode of electronic communication. We are also constantly working to build better and new functionality in the Orion user interface. However, the back end of Orion and the way in which we design our calculation methodologies are what give Orion its true superpowers. Orion uses a scheduler to dynamically spin up near limitless instances from Amazon Web Services to power calculations, which, where possible, allow for the distribution of data across instances in parallel to drastically decrease the amount of time that complex calculations at scale consume. It's important to note that while this architecture can handle massive jobs, it can also handle smaller scale jobs just as easily. Orion as a platform is designed to carry out any type of calculation relevant to the drug discovery process, and on top of the platform is layered a number of suites that can be licensed. The bulk of our science is found in the Small Molecule Discovery Suite, which ships with Orion and is the topic of today's talk. There are several other suites which can be licensed and are in development, but these will be left for another day. We'll start by taking a look at Orion's hit finding and identification capabilities, which center on the ability to carry out virtual screening of compound collections of any size. Virtual screening methods can be considered to be based on similarity to known ligands, known as ligand-based virtual screening, or based on complementarity to a known active site, which is known as structure-based virtual screening. At OpenEye, we use well-established methodologies to do these screens, a product known as ROCS for 3D virtual screening, and the Fred hybrid docking engine for structure-based screening. Before we get into how we screen, though, we need to touch on what we screen. Orion's parallel compute architecture allows us to take make-on-demand libraries and catalogs from vendors and create multi-conformer libraries prepared for input into our virtual screening protocols by using our conformer search algorithm, Omega, as well as our other cheminformatics solutions. Among the collections that we provide are compound collections from Enamine Real, Wuxi Galaxy, and MQL, which contain hundreds of millions to billions of compounds ready to screen in 3 dimensions. Of course, we also share the flow for preparation of these collections on the platform so that users can create their own screening collections from any source, as well as a flow for filtering these collections on a variety of cheminformatics criteria, including calculated properties, structural feature counts, or substructure. These screening collections can then be deployed in massive virtual screening campaigns. For ligand-based virtual screening, OpenEye created an application called ROCS, standing for Rapid Overlay of Chemical Structures, and its faster, closely related cousin, fast rocks, for performing rapid comparisons of molecular shape and features relevant to chemical binding, including acceptors, donors, rings, cations, and more. Scores are typically reported as a Tanimoto combo, which measures the degree to which shape and color overlap when the alignment of a query molecule and the best overlaid conformer of a molecule from the database are aligned. Higher Tanimoto combos have a higher probability of being isoactive against a particular target, and queries can be customized and iterated on to hone in on different features hypothesized to influence binding. Orion offers a couple of different ways with which to carry out such a search. The FastRock service on Orion allows for a user to quickly query a standing database of compounds, such as the default 50 million diverse subset of enamine real that comes preloaded on the SAS implementation of Orion. Shown on the left side of the slide is such a 3D similarity search, and at some point in the screen grab, you will have noticed the progress bar take a jump, and that jump corresponds to 60 seconds of real time. The results from this 3D search are then communicated clearly in a report and can be easily exported into a project on Orion. Alternatively, users can launch the FastRocks flow on any of our pre-generated vendor collections or their own bespoke collections to screen a database of their choice. This process is incredibly scalable, and searching the current enamine real collection consisting of 7.7 billion compounds that we have up on Orion takes about an hour and costs about $100. This is remarkable considering that Orion is performing almost 1 trillion overlays in this time period. Of course, if you want to do a more basic similarity search, this is also possible using a tool that ships alongside Orion, known as Molecules as a Service, or MAS. Unlike the previous video that I showed you, the search that I demonstrate on this slide is being done in real time using circular fingerprints against a 500 million compound subset of the enamine real collection. This search is done in about 2 seconds, and compounds again can be selected and sent into an Orion dataset for further modeling and manipulation. MAS also allows users to perform substructure searches of loaded databases, among other tasks. On the structure-based screening side, OpenEye has implemented its OE docking suite on Orion, which leverages the FRED docking engine for virtual screening. FRED is an algorithm that populates a defined active site with many translational and rotational states of each provided conformer of a molecule from a database, filters out clashes and other poor poses, and scores them against a scoring grid using the ChemGauss4 scoring function. In our validation and benchmarking efforts, we have found that FRED is among the most accurate docking engines for reproducing crystal structure poses and is certainly one of the fastest available docking engines. OE docking also ships with Hybrid, which fundamentally runs FRED as a docking engine, but uses information about a cognate ligand to help refine poses that will be evaluated downstream. Hybrid also has a multi-receptor mode, which will look at multiple structures of a given target with cognate ligands bound, and selects a docking receptor on the basis of ligand 3D similarity to the incoming molecule to be docked, helping to account for receptor flexibility. Orion also has many tools to support such a virtual screening campaign beyond just the docking engine. It has an implementation of our application Spruce, which can be used to prepare a protein structure from the PDB or prepare your own in-house structures. It also allows for import from our repository of pre-prepared structures, known as MMDS, or the Macromolecular Data Service. And this is accompanied by the GigaDock flow, which is our implementation of our docking engine on Orion. Just as with the classic OE docking tools, GigaDock allows the user to run a docking job at any scale using FRED, Hybrid, or multi-receptor mode. The flow is optimized to handle the enormous amount of I/O that accompanies the computational heavy lifting required of processing up to billions of molecules. Given Orion's ability to grab any number of instances from the cloud, it can complete a docking job at this scale in about 24 hours, though at a larger cost than ligand-based virtual screening. This also happens at a cost proportional to the size of the active site being docked into. Orion also offers a convenient 3D viewer for visual inspection of the docking poses generated, which also includes a spreadsheet view for looking at individual records and associated metadata. To close the section of the talk that focuses on virtual screening, however, it's important to note that the flow-based architecture of calculations on Orion enable us to combine both structure and ligand-based virtual screening in order to find new ways to intelligently explore chemical space. Two such recent methods that OpenEye has put together are FastRocks Plus, which does a ligand-based screen using FastRocks and then docks the top-scoring compounds into a provided structure, while also generating consensus and novelty hit lists. Another alternative is a GigaDock Warp, which docks a sizable subset of a given collection, identifying shape and color features important to binding. The top docked molecules are then used as FastRocks queries against the entire collection, to identify other compounds that have a high probability of activity, and those compounds are then docked into the active site. This flow has been found in our benchmarking to cost approximately 1/8 of a full gigadock run, while still affording about 70% of common hits to the full run. While Orion has certainly built a reputation as a powerhouse platform for virtual screening, the platform is also tailor-made to enable lead optimization campaigns. At this stage of the drug discovery process, teams are typically concerned with balancing several properties of a lead series in order to arrive at a candidate substrate for nomination into the clinic. These can include not only affinity for the target of interest, but also permeability, solubility, selectivity, toxicity, and more. The lead optimization process is inherently cyclical, and these sequential cycles consume time and precious resources. As such, priorities in these campaigns shift with time, and tools for overcoming each of these challenges is vital. Orion thus contains and enables a number of methods suitable for facilitating and accelerating the design phase of the lead optimization process, which we will cover in more detail throughout the remainder of the talk today. For those who are already familiar with OpenEye's tools for lead optimization, you will be happy to know that all of OpenEye's classic applications have been enabled on Orion. In addition to the docking and 3D similarity tools that we discussed previously, Brood, a 3D fragment replacement tool for finding novel bioisosteres, GamePlan, a tool for suggesting improvements to a ligand on the basis of water thermodynamics within an active site, Posit, a tool for posing new ideas within a lead series, Eon, a tool for assessing 3D electrostatic similarity, Freeform, a tool for ligand strain analysis, and SiteHopper, a tool for 3D binding site comparison, all have been enabled on the Orion platform to allow new and experienced application users access to these workflows in the cloud environment of Orion. Beyond enabling our traditional science, however, the massively parallel compute environment facilitated by the Orion platform also gives us a number of advantages when it comes to the development of new tools for lead optimization. These include methods for generating virtual libraries of chemical matter for evaluation, building of machine learning models in parallel, methods that harness the power of parallel simulation, and along these lines, predicting affinity of compounds using parallelizable molecular dynamics simulations. We envision that these methods can be deployed in sequence as part of a lead optimization cycle, where portions of chemical space can be enumerated to create a large but tractable number of ideas to be evaluated. And this list can be refined first with lighter methods, such as filtering, clustering, docking, and so forth. As the list of compounds shrinks, heavier methods relying on molecular dynamics, such as post-testing or relative binding free energy calculations, can be deployed, ultimately creating a list of candidate molecules for synthesis in a particular lead optimization cycle. Though it is important to note that the many tools for lead optimization on Orion can be mixed and matched at the will of the user, and this is not a prescriptive workflow. Orion contains a number of tools for kicking this process off, whereby a single idea can be expanded into many compounds for evaluation by a number of different methods. At OpenEye, we think about library generation on a sort of two-dimensional plane, where both novelty and accessibility of compounds is important in different circumstances during lead optimization. As a result, we provide tools that span the spectrum of use cases. For querying known chemical space, the previously mentioned FastRox and MOS tools are fantastic for searching for 2D and 3D similars from vendor make-on-demand or in-house collections of compounds. Brood, on the other hand, performs 3D fragment replacements and will create highly novel compounds, though with structures that aren't always easy to synthesize. On Orion, we've also developed other tools to explore virtual chemical space, with algorithms available to sprout atoms around a molecule, graft on fragments, or do replacements of portions of compounds using matched molecular pair databases. Most recently, we have been working on expanding the capabilities of our reaction-based enumeration engine, which builds new analogs of provided scaffolds using catalogs of provided building blocks and robust definitions for common reactions deployed in medicinal chemistry labs, giving users access to useful analogs that are likely to be synthesizable. These libraries can be filtered using a standard OpenEye filter file on Orion. However, for those who seek to do more dynamic filtering on the fly, datasets can be manipulated directly on the Orion Analyze page. The page comes pre-populated with a number of properties that can be calculated and appended to a dataset, as well as a plot window that can be adjusted for a variety of visualization tasks, and a number of controls for filtering on properties, other fields on the dataset of interest, or on the basis of substructure. These filters can be applied through the menus at the top of the Analyze page, and subsets of the data can easily be saved to new datasets for manipulation within Orion. Another common and fundamental task that can be aided by the parallel computation available on Orion is clustering, either to group similar molecules within a dataset or to select a diverse subset of a dataset for further analysis. For this task, we have a large array of flows that can aid in grouping compounds together on the basis of both 2D and 3D features. This newly updated package of flows contains methods for hierarchical clustering, clustering using K-medioids or dbScan, and also offers large-scale implementation of directed sphere exclusion methods in order to cluster up to 100,000 compounds. We've also recently released a flow package for building machine learning models for property prediction. The model building flow is capable of training models on the basis of the provided molecules, or if you prefer, custom provided feature vectors in tandem with some provided response property. The flows can build either regression or classification models, and will explore a variety of fingerprint types and featurization parameters, as well as machine learning hyperparameter options in parallel. The flows will also provide the user with metrics and actionable information on each model trained. Importantly, each of these models is built using LIMES, which enables the output models to provide an assessment of confidence in the predictions that they generate, as well as containing an explainer that will highlight atoms or fragments of a molecule that make contributions to a particular property prediction. We've also taken a particular interest in the power of parallel molecular dynamics simulations, leveraging the WestPaw toolkit. This powerful toolkit allows the simulation of rare events by carefully spawning multiple molecular dynamics simulations at various points of a trajectory, especially those where the simulation is making progress towards a particular objective. By keeping track of the weight assigned to each child simulation, one can glean kinetic data about a system in simulation, as well as observe critical mechanisms at play within the simulation system. Our first foray into this space is an intensive passive permeability simulation, which provides not just an estimate of passive permeability, but also gives insight into how these membrane-crossing events may be occurring. The hope is that these simulations will allow users attempting to rescue a lead series ideas on how to improve the permeability of a compound based on its dynamic behaviors as it enters, traverses, and exits a membrane. This work was recently published in JCIM and can be found in the reference at the bottom of this slide. With this development experience under our belts, we are also looking to take these parallel simulations into other, new, and exciting directions, and stay tuned for more updates on this front in the very near future. We utilize parallel molecular dynamics in other ways on Orion as well. Beyond allowing users to launch vanilla MD runs in the cloud, we offer an out-of-the-box solution for pose testing known as short trajectory molecular dynamics, or STMD. This method takes a posed molecule in an active site as input and runs a quick 6 nanosecond simulation on each provided ligand by default. The flow then takes a trajectory, clusters poses from the duration of the run, and scores each pose with the MMPBSA scoring function, as well as providing a knowledge-based score for each compound on the basis of observed binding interactions, which we call the BINT score. Scores are also created for pose stability, among many other metrics that users can use in their assessment of a candidate compound. And finally, we've been hard at work implementing and optimizing our OE affinity package. This set of flows at the moment is used for relative binding free energy calculations, but rather than use the more common FEP protocols, we utilize a method called non-equilibrium switching. This method utilizes heavy endpoint sampling and fast alchemical transformations in order to arrive at reliable data, instead of using one long alchemical transformation. The advantage here is the ability to parallelize calculations. Given that the flow assesses relative binding free energy, it is important to generate quality edge maps in advance. For this purpose, we have also created the OE mapper, which uses both 2D graph and 3D rock similarity to create quality edges in advance of the relative binding free energy calculation. When used in tandem, these flows can give reliable relative binding free energy assessments in hours, not days, and with substantial cost savings as well. To conclude, the purpose of this talk was to give you just a quick peek at the myriad of solutions that are available on Orion's small molecule discovery suite for use in early stage drug discovery. I hope that I was able to convey to you that the platform provides a user-friendly interface, but that underneath the surface is a powerful engine for running our well-known and trusted computational solutions at any scale, allowing the assessment of anywhere from tens to billions of molecules. Additionally, we provide data for screening campaigns to allow you to run actionable screens at the scale of your choice, while also providing you the flexibility to create your own bespoke screening collections to search just the portions of chemical space that is relevant to your particular project. But beyond this, Orion is not just for virtual screening. We've generated a whole new generation of tools relevant to lead optimization to complement and enhance the tools that we were already known for and have already made available on Orion, and include vital algorithms ranging from fundamental cheminformatics tasks all the way to relative binding free energy calculations. And finally, we feel that we have just scratched the surface of what parallel computation can bring to computational chemistry for drug discovery. And we are constantly adding new science to the platform to change the way that you do your work. And please, if you're interested in a deeper dive into the platform or any of the tools that you've seen here today, get in touch with us. We're happy to talk to you more about how each tool can be used, about underlying theory, or the validation that we have for these methods. And of course, thank you all for your time and attention today.
Welcome to the live session here, asking questions and answering some of the Q&A that we got in. David, thank you very much.
Absolutely.
And reminder for everybody, if you have questions, please type them in the Q&A section on your screen, so we'll get them and we'll get to answer them. I do see a few already, and there is one that says, do you offer any solution for quantum mechanics or DFT calculations on Orion?
Yeah, we do. So there's flows that leverage Psi4, and that's part of the standard Orion small molecule suite and also part of the formulation suite. On top of that, we also offer a Gaussian module that contains flows that can do things like torsion scanning, generating conformer ensembles, single point energy calculations. And then with the Gaussian module too, you can load in your own Gaussian input files to run on cloud resources. That requires a Gaussian license on top of the Orion license as well. So if that's something you're interested in, reach out, let us know.
Excellent. Another one. Oh, that's an interesting one. I think you show a lot of methods that we implemented, but question is, can I upload my own methods to Orion?
Yeah, so sort of. So the small molecule suite contains a bunch of out-of-the-box methods that you can run in the cloud. And of course, there are a myriad of ways that you can change the parameters in those flows to customize them, just as you would expect with our applications and other tools. You can modify the flows by adjusting some of the cubes within the flows, but it's not always a trivial process to get the cubes to align right. But to really customize the platform and the methods on it, or if you want to develop your own methods, you have to license a managed service from us, which goes beyond the SaaS implementation that we offer as a turnkey solution that we've talked about here today. But organizations that license a managed service, they get their own dedicated stack that's separate from the SaaS or multi-tenant stack that we offer. And that also comes with a command line interface to interact with the platform. So it can be a really powerful enterprise solution for customers.
Yeah. There actually was another question that just came in, and that says, what is the licensing scheme per seat, package, and compute, and how compute cost is organized? So if you want to get on, yes, it is a per seat license. So you, as a per user login that you get onto the platform. And the compute cost is actually prepayment. You would prepay the Amazon cost for compute in the end, and then we basically deduct from your account. That's one that just came in. What else do we have here? There is around money again. Do users get charged for the databases that they use in Orion?
Not for the databases that they use in Orion. So the databases that we talked about in the webinar, we cover the cost for generating and storing those fast rocks and gigadocking collections on Orion, which is a pretty substantial cost savings when you're talking about enumerating conformers for billions of compounds. We also offer stock databases for a number of the other flows that are on Orion. So like we offer patch databases for SiteHopper, fragment databases for Brood, and so on. We try and provide some turnkey databases for most of the methods that we have on the platform.
Next one. Can I get my data off the cloud, or am I locked into the Orion environment? Well, I hope it's not, but you probably can tell.
No, you're not locked in the Orion environment. I mean, we try and make it one-stop shopping for doing all of the molecular modeling tasks and analysis that you want to do. But we certainly understand that customers would want to do things with their data that aren't on the platform. So you can certainly download your data off the cloud to do things like trajectory analysis if you're doing MD runs on the platform, or local data analysis, or to transfer your data to a different platform or tool that you're using.
Yeah. We know that data is not only in Orion, and yes, you can get it off, I guess. There is another one. What kinds of integration do you offer? I guess that is an integration of other tools, or well, how Orion can talk to other things.
Yeah, so broadly within the methods that are on Orion right now, we've integrated a number of open source software packages into our methods. So like scikit-learn is used in places, GROMACS, Sci4, OpenMM. They're all integrated in different ways across the flow packages. If you have any specific questions about what is integrated or what you'd like to see, reach out and let us know, and we're happy to answer questions about what you can and can't integrate easily. Managed Services of Orion offers users really the opportunity to integrate bespoke methods of their own that they've developed for the platform, and the platform into their overall environment and workflows in very flexible ways. And we have a scientific integration team here that can talk to you about the various ways that you can develop on and leverage the platform for your unique needs.
And I guess those would be reaching out into in-house databases to bring the data into the platform and correlate or bring all the data together, small molecules and measured data that you have in your databases sitting there. So I think in order to stick to the time that we have allocated and be respectful of that for everybody, thanks for joining. All the other questions that came in, we will answer by email later today, I guess. I want to use the opportunity to remind everybody of our next webinar that is coming up. That actually is on SHMEP and game plan. So more focused on one of our tools to optimize your lead molecules by understanding water. So learning from water, what you can do to your small molecules to make them better. For today, David, thank you very much for preparing and giving us this session. For everybody dialing in and listening, thanks a lot. And I hope to see you at some other webinar or at a conference somewhere. Come and find us. Thank you.
Awesome. Thank you so much for coming.